Okay, perfect. Then welcome to this VPH honorary lecture of the fifth VPH summer school. And it's my great pleasure to welcome Alfons Hoekstra, who has accepted and to give this very special talk. So Alfons is a full professor of computational science and engineering, and he's director of the Informatics Institute at the University of Amsterdam. He's member of the Computational Science Lab, and he is leading several large European projects. So Alfons is a specialist in large HPC, so high performance computing infrastructures, where he has developed different types of heterogeneous simulations based on multiscale modeling and multiphysics and complex modeling. So I guess that if I remember correctly, you have also set up a whole infrastructure, so a framework, and to automatically then couple heterogeneous models and multiscale modeling frameworks, and which is actually quite unique. So if we're referring then to HPC infrastructures. So without further delay, Alfons, I'll leave you the floor, and thank you again for being here.
Yes, so thank you very much, Jérôme. It's really a great pleasure to be here. Of course, it is called a summer school. Well, I can tell you here in Amsterdam, it really feels like summer. So it's in a way a shame to be inside because, you know, the Netherlands is luckily slowly coming out of a lockdown, and I can tell you it would be great to be out and visit a little terrace and drink to the success of your summer school. Of course, it would have been so much better to actually be in Barcelona all together. I see many familiar names in the audience, and I would have loved to shake hands and say hello, but more important, I see many names that I don't know yet, and I would have loved to meet some of you in person. That's of course the whole point of being together, and I'm afraid, well, next year this will happen, I hope. So we're optimistic and positive about that, and yet I do want to thank Jérôme and his colleagues to push forward the summer school this time, be it in a virtual form. I think it's also very important, especially for, you know, PhD students that have just started, to be able to jump on an occasion like this one and to meet and interact. So thank you very much for doing that. I feel a little bit ashamed because I did not have more than enough time to prepare what I want to tell you. So I will explain a little bit about work that we have done on multiscale modeling and things that I believe could be of interest to you. Let's put it like that. I thought to not speak too much about high performance computing. That's if you're interested in that, feel free to contact me. I guess I can share my screen and then I'll, let me see. Yeah, that's possible. And then I'll start my lecture. So could you confirm that you can see it and that you can hear?
Yeah, perfect.
Yes.
Wonderful. So because then it's a little bit, I need to click away now. Yeah. All right. So Alfons Hoekstra, thank you very much for the introduction. I work at the Computational Science Lab of the University of Amsterdam, and currently I'm also the director of the Institute for Informatics. So whenever you're in Amsterdam, you know, for work or leisure, feel free to contact me, and myself and my colleagues are more than happy to welcome you in our city and our university. So, you know, before we start, I'm going to tell you lots of results of what I believe is interesting research, which is of course a joint venture of many people. And, you know, I didn't want to produce names because then I would definitely forget a few people. I just wanted to acknowledge all current and previous co-workers in the Computational Science Lab in Amsterdam, and many, many collaborators in these wonderful EU projects that bring us together. And let me see, here you see a few of them mentioned. Just to confirm, I'm moving my mouse. Do you see something?
Yes.
Yeah, good. Okay, so I can use that as a pointer. Thank you.
Well.
Good. Before we kick off, I want you to read this beautiful quote from Robert Pirsig from his, well, at least for me, famous book, Zen and the Art of Motorcycle Maintenance. He wrote somewhere, "The real purpose of the scientific method is to discover that nature has not misled you into thinking you know something you don't actually know." You should know that my background is in physics, and I like that quote a lot when I was studying solid state physics and I don't know what. But then I moved into biophysics and slowly into biomedicine. And at some point, I reread this quote, and this is so true, especially when, you know, from a more physics or engineering background, you try to make sense of physiology. I always find myself believing that I understand the systems that I'm looking at, but this is actually not the case. And, you know, let's try to somehow do some justice to this quote and keep asking how and why, and do we truly understand these very complex biological systems that, you know, one way or the other we try to mimic as models in computers. And to me, this is still amazing that we can actually do that. And it's also a great success, I would say, of the endeavor that we call the Virtual Physiological Human, that in fact, parts of nature we do understand to a certain degree. And we also understand which parts we don't understand. So that's real progress. So the plan of the talk is the following. I want to set the scene and, you know, be a little bit more generic than VPH, and speak a little bit about what I call actionable computational science. And then I want to zoom in to what we've called the virtual artery, and just talking you through one or two applications, depending on the amount of time that I have. I do want to touch upon a very exciting project that we are currently doing, and I've seen some co-workers of that project also in the list of participants today, on in silico stroke trials, and then I want to wrap up. So unfortunately today I'm not going to speak about multiscale computing or high performance computing, but again, as I said, if you find that interesting, do contact me, and we can have some private chats on that. So computational science. This is a definition that I wrote myself, and you may or may not agree to it, but I believe this is all about formulating computational models of phenomena that we observe, and then to perform computer simulations, to perform an experiment on your computational model. And why would we do that? Well, first, to deepen our understanding of the systems that underpin the phenomena. And then once we have this understanding, we would be able to predict their future behavior, or to find adaptations that would change the behavior of the systems in some desired way. And this has quite some deep implications. And this is for me the starting point of all the modeling and simulation that we do. It helps us to better understand systems that we study, and we may even be able to predict their behavior or change their behavior in some way. And so how would that look like? And I believe that there is a modeling and simulation cycle that we all go through when we are in this field of computational biomedicine. And so it all starts with systems that we study, and we typically have some, or when you're lucky, lots of data available, some or lots of theory. And from that, we usually build a conceptual model where we say, listen, we have this tissue that is strained, and that may lead maybe to some biological reaction. You know, in words, you build conceptual models based on that. And we quickly, immediately translate that, doing quantitative modeling, to a computational model. So that could either mean that you write down equations that you want to solve, or if you're, for instance, into agent-based modeling or things like that, you would maybe write down rule sets for these agents. And then we want to perform experiments on the computational model, and that is what we call a simulation. And that usually implies to put the thing on a computer, to discretize it, to implement it. And then we can actually run experiments on the computational model, and then we can close the loop, because we can then compare to the data that we may have on of the systems that we study in order to, for instance, validate the computational model, or we could start to predict things, test hypotheses, change the systems, etc., etc. Now, I challenge you this week. I'm absolutely sure that you will be going through this cycle over and over again, or you may find yourself spending lots of times in one of these boxes. And this is just how we work in computational science. And you could say, let's look at an example. This is one of my favorites. I'm very much interested in studying the behavior of flowing blood at the cellular level. So what individual red blood cells and platelets do. And here you see a beautiful movie where down here, you see this beautiful cell-free layer, and you see some red blood cells rolling and tilting and doing interesting stuff. And then here you see a two-dimensional simulation of the same thing, where we try to mimic what actually happens in here. So if we would wrap that to the cycle I just discussed, the real phenomenon is flow of red blood cells and platelets. We want to model that in some way. And in this case, we have a mathematical model. I'm not going to speak about the details today. We need to discretize and implement this. And if you really want to do this in 3 dimensions, well, you do need big computers. Here you see a very big one that is installed in the Netherlands, but you could also build some less heavy models, and that would easily run on your laptop. Then we can simulate our model, we can analyze the results and close this pipeline. And so this is for a computational scientist, a very important mode of working. And I guess it's also interesting and important to understand where you are in the activities, in your daily activities. But there's of course much more. We are not the only ones from a computational point of view that study systems. Of course, we have our friends in the wet labs, in the field labs, etc. We have our simulation-based science laboratory, and we all collaborate. I guess many of us would be here, you know, behind the computer in a simulation-based science laboratory. But some of us also work in these labs or have very important collaborations with people that actually study the real systems in vivo or in vitro and produce data. What I find most interesting is if we are able to get into a true collaboration with our colleagues that are either in the clinics or in the labs, and work very close together, even to the point that you may also be doing some experiments yourself or engage in clinical studies or what have you. That has been extremely good for the progress in our field, and we have to keep doing that. So then I want to put the word actionable in front of computational science. We want to do this for a purpose. If we are able to do predictions, then that should lead to some action. For instance, you may want to design something. You may want to design an improved stent for coronary artery stenting. It may lead to decision support, where you may want to help decide on the best treatment for a specific patient, or maybe you want to help in designing clinical trials. And this is the example of the INSIST project that is about in silico trials for treatment of acute ischemic strokes. So you may want to help to design real trials by first running designs in silico, and in that way speed up the actual development of better treatments. In order to do so, in order to be able to make your simulations actionable, that requires that your computational models are reliable and trustworthy. And you know, that immediately means that we have to work on verification, validation, sensitivity analysis, and uncertainty quantification. And, 10 years ago in the field in our community, there were not so many people doing this in a very systematic way. It is very good to see that over the last few years, this is becoming a standard part of our studies, to do the VVUQ and the sensitivity analysis. If you want to read up on that, I can recommend these two books here, Verification and Validation in Scientific Computing by Oberkampf, and the book by Smith on uncertainty quantifications. These are pretty good introductions to these fields. I would really hope that any simulation that we do is somehow also taking into account uncertainty quantification, sensitivity analysis, etc. And why is that? Because I want to show you another quote by Rumsfeld, the former Minister of Defense from a long time ago in the States. And he had this beautiful quote, which we can apply to our field if you want to do UQ, uncertainty quantification. And the quote is as follows: as we know, there are known knowns. There are things we know. Okay, that's the knowledge we know. That's good. We also know there are known unknowns. That could be, for instance, some uncertainties on parameters that you use in your model. You know that you do not actually know to a very high degree of certainty the value of that parameter, but you may know a statistical distribution where you would find these parameters. That is to say, we know there are some things we don't know. That's also very important because that we can express one way or the other in our models. However, the nasty thing is, and that's also back to the quote of Pirsig, there are also unknown unknowns. The ones we don't know. These are the nasty ones. And that's why we always have to be very careful in the modeling that we do, especially for this modeling physiology, pathophysiology. There will always be unknown unknowns, would be my statement. And we have to be very sensitive to that by constantly going back to the actual system, consulting actual data coming from the actual system, and being very, very critical at the modeling that we do. So I want to make a few more generic statements, and then we jump into more VPH related stuff. This is another beautiful quote that I want you to, somehow, well, not completely remember, but remember the summary. What are the strengths and weaknesses of simulations, of the things that we do? It's the generality and the ease of the application. And now here's a quote from the book of François Sellier. All too often, simulation is a love story with an unhappy ending. We create a model of a system and then fall in love with it, which makes sense. Typically, we work for many, many years on our favorite model, etc., etc. Well, we all know love is usually blind. We immediately forget all about the experimental frame. We forget that this is not the real world, but that it represents the world only under a very limited set of experimental conditions. We become model addicts. And currently we use the phrasing context of use. We have to be very, very clear on the context of use of our models, and sometimes we forget that. So the way to remember this is the following. It's very simple. Don't fall in love with your model. And we know this is hard, but each time you could say it to your office mate, hey guy or hey lady, don't fall in love with your model. Okay. A few more things on verification and validation. I'm not going to talk you through this picture, but just to be sure what we mean, verification is the process of determining that the way we have implemented and discretized our model actually represent the conceptual description of that model. And validating is about determining to which degree the model is actually representing the system under study. And you could paraphrase that in the following way. In verification, are we solving the equations correctly? Which you could say is an exercise in mathematics and computer science. And validation is, are we solving the correct equations? Which is an exercise in modeling. And again, in order to make our simulations actionable, we have to be very, very clear on these concepts and these ideas. Now, I'm not going to do this. In the context of the Virtual Physiological Human, and many of you may have heard of this, we have this AMSC V&V40 document assessing credibility of computational modeling through verification and validation, and then specifically applied to medical devices. If you're in this field, I would say this is an important document if you want to make your models and your simulations actionable, but this goes much further. It is also about credibility of your models. I could very much recommend this paper by Marco Viciconte and some of his colleagues that was recently published on the verification, validation, and UQ of predictive models used in a regulatory evaluation of biomedical products. It basically gives you, on one hand, a nice summary of this V&V40, but also reflects a little bit on the limitations of the V&V40 and what it could mean in other contexts and for other types of models. This is a very good read. And if you find some time this week, you may want to jump on it. Okay, then I want to go to a topic that is very much researched in our computational science lab, which is what we have called the virtual artery. Of course, this is a multiscale model, and you are fully aware of this, where we look at our system at many different scales, and then we apply this idea of the middle out approach. So if we're interested, say, on the behavior of an organ, we may be taking into account some lower level scales like the tissue scale or the cellular scales, and maybe some upper level scale, the organ systems, and then we close our models. Now, in the if we look at models for our vasculature, we can also identify a number of these scales. We have the full body scale, which we typically model using one-dimensional models. Then we're down to the, at some point, the millimeter, centimeter scale of, say, arteries, where we would be doing full-blown three-dimensional time-dependent modeling of flows. We are very much interested in cellular models, for instance, looking at the cellular details of the arterial wall, or looking at blood as a suspension of red blood cells and platelets and leukocytes. You could also go subcellular, so look at molecular processes. And of course, you want to couple these things together. There is information flowing from the cellular level. The behavior of the red blood cell, in a way, determines the rheology of the flowing blood. You may need to take that into account in very complex flow situations, and we have lots of examples of that. So we have organized this multi-scale model, and this is something you see a lot in what I call a scale separation map. So we have identified temporal scales here, and we have spatial scales on the horizontal axis, and then we write these boxes that represent processes on these scales. And when during the introduction, Jérôme talked about this formal framework for multiscale modeling, we have done lots of work on formalizing this cartoon way of expressing a multiscale model into a mathematical construction, and also into software frameworks that allow you to glue models together in a very easy way, and also into execution frameworks that make it much easier to run these multiscale simulations on all kinds of hardware. Again, I'm not going to talk about that, but so there's lots of deeper thinking behind this idea of a scale separation map. Now, so the virtual artery, just again to set the scene, this is going to be a multiscale model for peripheral blood flow, from whole body blood flow down to single cell transport. And it should, when needed and where needed, relevant details of biological, biophysical, and biochemical processes also happening on many scales. And if we do that in a right way, we can study fundamental curiosity-driven questions. For instance, why do platelet marginate? We know that they marginate, so they move to the vessel wall as blood flows, but the exact biophysical reasons why that is are still not fully understood. And there is even more interesting stuff going on. For instance, leukocytes, white blood cells, are also able to marginate, and that is not a complete physics-driven process. There's also biology going on. It seems that these leukocytes are able to actively change their stiffness, and that which is a biological process, and that would induce stronger margination or not. So there are lots of exciting, interesting things going on. But you could also apply this, of course, to improve treatments or medical devices. And many of you are actually working in that field and have lots of experience with that. And I will talk about the stenting of coronary arteries. But you could also try to then maybe predict rupture of intracranial aneurysms. Or you could aid in silico clinical trials. And then what you would probably be doing is create relevant samples of a certain population and then study hemodynamics in some way in that population. You could also think of personalized medicine applications where you would then create a relevant sample of physiological conditions for an individual. So you're still sampling, but now for one individual, and maybe again study hemodynamics, well, etc., etc. And so how would that then look like? And I'm going to show you some cartoons of what we are implementing right now. And so we have whole body models, and these are typically 1D or 0D models of blood flow, and you may have seen these models in action. And so here we see an example of a full body model that we have developed in Amsterdam. But then we want to couple those to 4-dimensional hemodynamics. And so you would be using the 1D or 0D models as boundary conditions. And for instance, here you see an example. But then we also want to couple, if needed, and in some applications it is actually needed, to cell-based models. For instance, cell-based models of the arterial wall and or cell-based models for the flow of blood. And I'm quickly clicking through, but here you see some examples that I will discuss in a minute in more detail. And here you see some nice movies of this stuff. So here we see just a simulation of systolic blood flow. Here we see some growth of neointima inside an artery, and here we see the flow of red blood cells and platelets. And you can imagine that it would be great if you study a certain part of the arterial system that you want to be able to plug in these types of models where and when needed. And we are working on that kind of technology. All right, I want to show you one extended example. This slide deck actually contains much more, but I won't have time to talk you through everything. So we're going to stick to this one example of In-Stent Restenosis. This is work that we have been doing for a pretty long time, and it has many, many of the ingredients that I just mentioned. So this is how it would look. This is how it looks like. We have coronary artery disease. So here we see an angiogram of the coronary arteries, and here we see a severe stenosis. And if you would look at these arteries, this is a very severely stenosed artery. Standard treatment is balloon angioplasty and then implementation of a stent as a scaffold to keep the vessel open. And this would be an image of a coronary artery severely stenosed before treatment, and this would be immediately after treatment. So we see a nicely reopened artery. Now, what is then in-stent restenosis? This is basically after you have opened up the artery, there could be a regrowth of tissue, basically the development of some scar tissue inside the lumen of the opened artery. If you look at these histological pictures, these black dots here are the struts of the stent. And then here, this grayish stuff here is some neointima, so newly formed tissue. And this is a beautiful healed artery, meaning that at some point, the lumen basically stabilizes, and this is what you want to see. However, this formation of neointima can also keep growing very fast and again start to occlude the artery. And that's what we call an in-stent restenosis. And in clinical practice, if the artery is again more than, I think it was 50% closed by neointima, it is called a restenosis that could give rise to additional treatments. Now, in the old days, there were so-called bare metal stents that suffered a lot of this in-stent restenosis, and then came drug-eluting stents and all kinds of other variants that basically were able to reduce this problem of in-stent restenosis. However, drug-eluting stents again also have their problems and complications. So if it is possible to administer a bare metal stent, then that would be great. We have started to see if we could mimic, build models for this process of in-stent restenosis, and with that, if we could then help in better designing stents or trying to predict on a cohort level or on a personalized level whether somebody would develop an in-stent restenosis or not. So let me talk you through that. And so what is in-stent restenosis? It is a maladaptive response of the coronary artery vessel wall after stenting. And the main challenges when we started to model this was to understand the details of this pathophysiology and basically to understand why it starts. So why would you have a neointima formation in the first place and why it stops? And would we be able to mimic that in a mathematical model? And once we have that validated, would we then be able to improve treatment via, for instance, improved designs of stents? All right. So the pathophysiology looks a little bit like this. We deploy a stent in a coronary artery. This leads to an arterial injury because basically the balloon angioplasty completely removes the inner layer, the endothelial layer in the coronary artery. And typically what also happens is that the next layer, the internal elastic lamina, is stretched or maybe even damaged and ruptured. This immediately leads to an inflammatory response. It leads to the activation of smooth muscle cells, and the V stands for vascular. So for vascular smooth muscle cells, they start to migrate into the lumen and they start to proliferate. They start to also formate lots of extracellular matrix, and that leads to neointimal hyperplasia. Now, this all started also with the availability of data from in vivo studies in pigs. These were collected by Julian Gunn at the University of Sheffield. And at that time, he had lots of samples of stented porcine arteries collected over a pretty long period. And the interesting thing was that this was basically a data set that was available. And when we started to our efforts to model in-stent restenosis, we found that that was pretty much a gold mine, and we started to mine that. We had to spend lots of effort to take the right information out of this data set and to use that for our purposes. And I believe we find ourselves on many occasions in such a situation that there is there are many collections of clinical or preclinical data sets that when you look at it with the eyes of a computational scientist, of a modeler, you may want to try to get other types of information out of those data sets than what they were used for originally. So reusing all this data for instance, for validating our models is really an important thing. And I think we should be doing that much more often than what we do now. Currently, 6 months ago, we started a project called the In Silico World Project, which is headed by Marco Viciconte, and many colleagues are part of that project, which is also, as part of that project, looking at data sets, curating data sets, making them available in a way that we can use them to validate our models. And I would very much hope that we, as a community, keep doing that. So what does this data set look like? As a function of so-called injury score, so that's basically how deep you push the stent into the arterial tissue. You see the amount of neointima formation, the neointimal thickness, as a function of time after stenting. And the data points we're taking at 14, 21, and 28 days. And mind you, every dot here basically means one animal. Now, if you translate that in a way that we as modelers like more, something that depends on time and is dynamic and changes over time, this is what we got. And this has been an important data set in our first trials to validate the models that we have. And so what you should see here is that with an increased injury score, as you go from injury score 0, the black line down here, to number 4, you see that the response is faster. And second, also, if you look at 21 days, you also find that the endpoint, the amount of neointima correlates with the injury score. If you wait a bit longer, you see so-called remodeling. It seems that the neointimal thickness becomes smaller again. That's a process that we did not model. So we basically model up to 21 days. All right, so the first thing we did was to formulate a multiscale model of in-stent restenosis. And what we did was a kind of group modeling effort where with some biologists and interventionists and cardiologists, vascular biologists, we were sitting together and we were basically naming all the relevant processes that the group thought were important for this process of in-stent restenosis. And this was from the subcellular level up to tissue level. We formulated details of the single scale models and we talked about how they would be coupled. And then we tried first in a conceptual way to model these things in detail. And this resulted in a very complex scale separation map. So it's very hard to read and well, maybe you don't want to read it, but this box here, which sits on a certain temporal scale down here and at a certain spatial scale up here, is the smooth muscle cell hyperplasia. So this is what gives rise to the actual neointima, but that is coupled in many different ways to physical processes, to biological processes, and in a complex way. We quickly realized that although this was an interesting way to organize the knowledge that we had on many details of the process, this would not lead to a simulation of these hyperplasia. That's just that was just too complex. So we simplified the model and we simplified it even more to this, which is something we can handle as a mathematical model. So we have the smooth muscle cell proliferation up here, which looks at a model of a cell cycle of a single vascular smooth muscle cell, and then at the proliferation of these cells. And that together leads to the neointima. This is coupled to blood flow because, as I will explain in a minute, the wall shear stress is important for the regulation of the smooth muscle cell proliferation. It is also linked to a diffusion process, and that has to do with these drug-eluting stents where drugs are diffusing from the surface of the stent struts into the tissue, and that inhibit also the smooth muscle cell proliferation. So this is what we have implemented. And again, I'm not going to speak about the details of this multi-scale model. I will mainly focus on the proliferation of the smooth muscle cells. All right. So first we need to understand or hypothesize on why this in-stent restenosis starts. So why do you get the proliferation of the smooth muscle cells in the first place? And what we have learned from the biologists and the specialists of smooth muscle cell biology is that the angioplasty and stenting leads to a change of the smooth muscle cells of, a resting contractile state to a synthetic vascular smooth muscle cell, where these cells start to migrate through fenestra in the internal elastic lamina inside the lumen. And once they're inside the lumen, they start to proliferate. And these are the processes that we actually try to capture in our model of the vascular wall, as I will show you in a minute. And so once in the lumen, they start to proliferate. Now, they are also regulated by endothelium. So these vascular smooth muscle cells can return to their contractile state and therefore stop growing. So that would stop the neointima formation through a number of pathways. And one important pathway that we have implemented in our models is through the production of nitric oxide by the functional endothelial cells. And that is, again, also depending on the wall shear stress as experienced by these endothelial cells. And down here, you see an example of how nitric oxide or nitrate production depends on this wall shear stress experienced. Now, as I told you a while back, because of the balloon angioplasty, the endothelium is completely gone. So once the smooth muscle cells start to proliferate, there's nothing to stop them. So there should be a process of re-endothelialization going on as well. And once the functional endothelium is there, and once the wall shear stress is in a range where there is sufficient NO production, then these vascular smooth muscle cells will stop. And this is the kind of detail that we had to implement into our models. All right. So the main ingredients of the agent-based model for the vessel wall, and this is something that that requires lots of computing as well. We model every individual vascular smooth muscle cell as an agent, and these agents interact. These agents can move, they can divide into daughter cells, etc., etc. And so the dark blue dots here are neointimal cells. So these are these vascular smooth muscle cells that are migrating and are growing. The brownish things here are the stent struts. We have the red stuff here is the external elastic lamina, and we have an internal elastic lamina, the bluest ones here. Remember, that's the lamina that could either rupture or have little holes in there through which the vascular smooth muscle cells can migrate into the lumen. If you put all that stuff together, you couple it to blood flow, and then you can mimic the growth of the of that neointimal tissue. And here you see an example. This is immediately after stenting. This is after 14 days. This is after 28 days. Okay, I'll quickly talk you through some results now. And so after stent deployment, that could look like this. 7 days later, we start to see some neointimal formation, and 28 days later, you see a restenosis developing. Now, what we have done is to run many simulations as a function of an injury score and up to 35 days. And first, when you look at it and compare to the data from Julian Gunn that I have copied again here, we see the same behavior. We see that as we increase the injury score, we see a faster response. After about 21 days, the response seems to level down. It is not going down again like here. The remodeling is not part of the model. So we were quite happy to see this. There is one little thing that is not correct. If you look at the actual thickness of the neointimal area, or of the neointima, which is 0.4 millimeters down here, you see that we're basically a factor 3 off. Because in the porcine system, it was in the order of 1 millimeter. This is due to the fact that in this model, we did not yet include production of extracellular matrix. I will skip this, and we needed to move on and add to the model the growth of extracellular matrix, and we also wanted to compare a much more detailed histological data. So I hope you realize we have now already gone 2 or 3 times through this modeling and simulation cycle. And we try to validate, and parts of the model are validated, parts of them are not yet validated. All right. The way we model this extracellular matrix production is by realizing that when we have synthetic smooth muscle cells, that when they are a little bit strained, this is again known from the biology of these cells, they start to produce extracellular matrix at a constant rate. And the way we have implemented that is to have a statistical process that with certain probability per hour, we produce an agent that has the same volume as the smooth muscle cell, but is extracellular matrix. So that's just an agent that is no longer growing or dividing. Now, if we put that in, we can get much better results. We have also much more detailed histological data to validate the model. And basically what happened again from the Julian Gunn data set, it was possible, and this is work done in close collaboration with Polimi in Milan, to get a complete and detailed picture of the stent as it was deployed, as well as histological sections along the stent after 14 and 28 days. And the good thing of that data set is we could also get an initial condition for the simulation. So we had from this 14 days or 28 days data set, we had an initial condition, so at day 0, and then follow-up data on 14 or 28 days. That was cool because then we could actually compare histology and compare our model with this in vivo data. And I don't have time to talk you through the details, but just to convince you that we are doing much, much better than in the earlier attempts. So if you, for instance, look at well, the graph here, red is our in silico production along the struts. So if you would count the struts here along this cross section, and you would actually measure the in vivo neo-intimal thickness and then run the simulations, you know, we are on the correct order of magnitude. We seem to be getting this, the histology pretty good. Of course, there are also parts where it is not that good, but we have a pretty good understanding that we are getting close. And this was very encouraging because putting in the extracellular matrix results in the correct amount of neo-intimal thickness. You can look at that in lots of detail. I won't do that now, but this was also a very interesting thing when we then looked at the neo-intimal area percentage, because from a clinical perspective, that is important. You know, whether you develop an in-stent restenosis or not is basically whether this neo-intimal area percentage is below a certain threshold or not. And here we are doing very, very good with our in silico model as compared to the in vivo model. Well, we have also performed uncertainty quantification. As I told you, if you want to make these things actionable, you actually have to do uncertainty quantification. And I won't talk you through the details, but here you see 2 examples of what that actually means. So here you see as a function of time, the development of the neo-intima expressed in the neo-intimal area. The black line here is the mean value, and then the reddish area here is the so-called total uncertainty. So what we have done is you actually sample over, in this case, 4 uncertain input parameters. You run a few thousand times the model, and then you create distributions. And of course, in terms of compute power needed, that is pretty expensive, as you can imagine. But what is even more interesting is if we would now at the endpoint, and that is this graph here on the right, if we would look at, for instance, this distribution at 28 days or so, then you find a distribution like the blue line. And around 0.4 neo-intimal area is the cutoff, that's this dotted line, is the cutoff between calling it an in-stent restenosis that may require additional clinical intervention or not. And then through this uncertainty quantification, we could actually then report a probability of an in-stent restenosis to happen. And that probability is basically the area under the curve after this 0.4, which in this example is in the order of 10%. Now, we haven't really looked at that in lots of detail, but we believe that by interpreting the result like that through the uncertainty quantification, we could actually, we would be able to extract probabilities of developing in-stent restenosis for a given design, for a cohort of patients or for a specific patient. So what are next steps? And I'm getting slowly to the hour, so I'll be wrapping up quickly. Next steps are to actually compare to human in vivo data. So far, we have validated our model, and we are still doing that, by the way. We're not satisfied yet on porcine data, on pig data. We are very happy to collaborate now through the InSilk project and the In Silico World project to also get access through our friends in Rotterdam to human in vivo data. And here you see a few first pictures, which is based on OCT. And here you see some OCT images, a pullback of a stented artery. And the kind of things that we then have are OCT images post-deployment and in follow-up studies. And this is in a human system. And currently we are working very hard to validate the models, but now for the human system. And based on this really unique data set. And it is a very complex system to try to model, but we believe that we are getting close to a point that we can claim validity of the models for the question that we want to ask to the model. And that is, what will be the neo-intimal area that develops over time. Let me see. So now I'm going to skip a shorter example because I don't have time for that. I do want to speak quickly about another application where we are much more into the 1D and 0D blood flow modeling, but to show you another end of the scale where there are also very exciting model developments where we try to glue different models together. And the context is in a project called INSIST, in silico trials for the treatment of acute ischemic stroke. The overall objective of this project, which is an EU-funded project, is to realize in silico stroke trials for acute ischemic strokes. And we do that by building populations of virtual stroke patients, to have in silico models of treatments, and to have in silico models of the human response to a stroke, and then to validate these models and to do some proof of concepts. What you need to know about treatments of acute ischemic strokes, that up to 2015, the standard treatment was intravenous thrombolysis. So getting a thrombolytic agent and just hoping that the clot would be removed by this thrombolytic agent. And that was not very effective in large vessel occlusions. In 2015, the Mr. Clean trial that was performed in the Netherlands showed for the very first time the effectiveness of mechanically removing the clot through so-called thrombectomy, where you go in with a stent retriever, these little these little struts here that basically capture the clot and pull it out. And this is how it would look like. This is before treatment. Here you see a clot freshly removed from the brain and, harvested. And we are very lucky to be able to actually study this fresh material in the lab. This is also done in Rotterdam. And we have learned a lot by the availability of these kinds of specimens. And this is what the clinicians hope to see after treatment, a very good reperfusion. Now, unfortunately, even if thrombectomy is successful, for many patients, the clinical outcome is not. And that triggers many more new trials to improve treatment of acute ischemic strokes. And we wanted to complement that with in silico trials in order to speed up and to better design new and running clinical trials. Now, I won't have time to talk you through the details, so I'm going to skip this. Here you see two nice movies of these treatment models. The thrombectomy model, where you see a clot actually being pulled out of the M1 and through the IKA. This is work done again in Polimi, Milan, really, really exciting work. And here you see an example of a simulation of thrombolysis. This is done at the University of Geneva, the group of Bastien Chopard. What I wanted to talk to you about, and if you allow me, I will take a few more minutes before I wrap up, is to model the actual brain perfusion and infarction. So how do you get blood to the brain? How is it perfusing into brain tissue? And can we mimic an infarction? And this is joint work between the University of Amsterdam and the University of Oxford, the group of Stephen Payne. And basically what we wanted to do is to be able to predict the location and the size of an infarct. And typically what we know is the architecture of the circle of Willis, and everything from the circle of Willis to the brain is basically not completely known, but yet we do want to mimic that. So what have we done? And again, it's all about scales in your model. We model the large arteries using a one-dimensional blood flow model. Here you see the circle of Willis. We attach to that data that is known about cerebral arteries. And at some point, we do not have imaging anymore. And then we construct bifurcating trees that are then mapped onto the pile surface, so the brain surface, and we link that model up to the so-called penetrating arteries that go deep in the brain. And then we have a three-dimensional perfusion model developed in Oxford. And we have to glue all these things together. Now, let's skip this. It's all beautiful stuff, but I don't have time. A very important part of this modeling is the fact that we have so-called collateral circuits. So if we experience a stroke and part of the brain is no longer receiving blood and oxygen, there are all kinds of secondary pathways that could still provide some blood to the parts of the brain that are in danger. And we know from clinical observation that these collaterals are extremely important to mimic this process of stroke and infarction. So that is also implemented in the model in a very detailed way. And so once we have done all these things, so we have created this fully coupled model of the larger arteries to the circle of Willis, to the cerebral arteries, coupling it to the pile surface, having the collaterals, and then coupling this to perfusion models, we are capable of predicting the reduction in perfusion after a stroke, and also as a function of the collaterals. So here you would have hardly any collaterals, and you see a pretty strong reduction of perfusion in a big part of the brain, and this would potentially lead to a huge infarction. And as we go to better and better collaterals, then we find this. We are currently in the process of taking clinical data on collateral scores, and with that parameterizing our collateral model. And again, this is an example of reusing lots of data that is known from clinical trials in thrombectomy and stroke treatment in order to parameterize and validate our models. And then in the end of the day, we are able to model the development of a stroke. And here you see an example. The black part is an infarcted part in the brain in our simulations. And here you see examples of actual stroke patients. Again, this is work done in Oxford to further validate these models. And well, I did want to show the INSIST consortium, a great group of people to work with, and fantastic work going on there. So I want to wrap up. Jérôme, I'm almost done. I think in 2-3 minutes, I've been talking for an hour, so that's more than enough. First of all, as you may imagine, the stuff that I've shown you and also the stuff that I did not show you is based on a huge collection of papers. And if you're interested in that, you may want to go to this link, and I can share that to you and just download this stuff for further study, or again, just to contact me. Okay, to go full circle and to wrap up, I just want to encourage you to keep asking how and why. This is what we did in this modeling of the in-stent restenosis. You know, I kept asking, and my co-workers, we kept asking to the biologists, to the vascular surgeons, to the specialist, yeah, but how does it work? And why does this happen? And why does that happen? And they say, hey, these are great questions. I have some data. I believe it is like this. And then we could make decisive and big steps in this cell-based modeling that we did. And of course, you know, to go back to Pirsig, let's keep asking how and why to make sure that nature has not misled us into thinking we know something that we actually don't know. So let's make sure that we keep doing this. Second, let's make our simulations actionable, always. So to the very least, please do a UQ, always, or to put it in a very colloquial way, put error bars on your simulations, but also really work on validation and verification and make that an intricate and standard part of any modeling and simulations that we do. I know how hard it is. I realize how hard it is, or that you may have hardly any access to data that you can use to validate. But then, hey, you know, keep talking to your colleagues, look a bit further. There's always data out there that you could use to make steps, or, you know, convince your friends and colleagues in the experimental labs to help you produce new data that could help you in your modeling efforts. And finally, you know, a very generic statement, but since this is in the VPH summer school, I do want to make it. Let's keep rolling out the vision of the VPH. I think what we are all doing is extremely exciting. And, you know, the last few years, I believe we are moving into a next phase, which is extremely rewarding and exciting, where we see true clinical applications of, let's say, 20 years of model development in the VPH, true clinical applications, in silico trials on the way, FDA and EMA actually approving what we are doing, the industry being extremely interested in the academic research that is done. And I think we have to do and pay credit to the Virtual Physiological Human Institute that has played a very important role in pushing us forward as a community. And Jérôme, with that, I would like to thank you for your attention, and I'm ready to take some questions.
Fantastic, Alfons. Thank you very much for this very great talk, and as a very inspiring and very useful message for our attendees. So I would like to invite for questions. As far as I can see, I don't see any question in the chat. So I don't know why the chat is closing permanently, constantly. So do we have any question? No. So maybe I can just start and break the ice. Yeah, I would like to ask you about when you're doing actually this scale separation, and so sometimes specifically at the cell scale, when you're working on multiscale modeling, it's very hard to have the time constants for the biological processes that you want to simulate. And in those cases, basically, or I don't know if you have then already an experience of a decision you had to make in order to place the time scales of your cell action within the time scales of the blood flows.
Yeah, I think that that is a very good observation, and it turns out that the example that I talked about today, mainly this in-stent restenosis, is very much determined by the time scale of the reappearance of the endothelial cells, and you know that is a big unknown, and that there is some literature on that. We were quite happy to see some recent experiments also done in Sheffield in the group of Evans, looking at migration and re-endothelialization also inside these cells. But that is a time scale that is measured in order 10 days, 10, 15, 20 days. We have the vascular smooth muscle cells that that have a cell cycle of order day. And so that means that and we know that this whole process, this whole biological process, it takes also, you know, order 3 weeks, in humans longer. So in the porcine system, it basically stabilizes after 3 weeks. And these are all long time scales, but also separated. So that already makes it hard. And a few of these time scales we don't know. On the other hand, you have pulsatile blood flow, which is order seconds, right? And we have to compute the wall shear stress. But that is locally well separated. So at every time step of the agent model, which runs at time steps of an order of an hour, we basically couple that to a single shot of a blood flow simulator, compute the wall shear stress, and feed that into the agent model. But the biological timescales, although they are in that case pretty close together, 1 hour to a few days or weeks, they do present lots of problems because we don't know them very accurately. And from sensitivity analysis, we know that that's one of the most important parameters that will determine whether you have an in-stent restenosis or not. If you go to other systems, so for instance, the example that I could not study, but we use also cell-based blood flow simulations. So looking at the red blood cells and platelets to study the initial aggregation of platelets in thrombogenic environments, and then all the processes that then lead to a thrombosis. These are also pretty fast processes that happen on the microsecond scales, these initial aggregations, but a full occlusive thrombi you typically measure in minutes, or 10 minutes or so. And that's a huge difference in timescales, and it's pretty hard to capture that in models. And hence, you make all kinds of assumptions, and you assume things constant where you know that they are not. So that's a known unknown. In a way, we're always quite happy if the timescales are separated, because then you can assume that you can average over the fast process and keep the slow process constant. When the timescales are still pretty much separated, but also they start to overlap, things become much more complex in the whole modeling. And especially, as you said, when you go into this world of cell-based modeling, you have to take that explicitly into account, and that's far from trivial.
Yeah, so I guess if I can read a little bit between the lines, so I guess in the end, and the key is to have the highest level of modeling as possible, so that you can match actually effective timescales according to what you're observing.
Yeah, but that very much depends on the question you want to ask to the model. So if you look again at the example that I talked about, it so happened that the process that we were mostly interested in, so the neointima formation, also lived on the longest time scale. But that is not necessarily the case. We all realize that in many ways we use this middle out approach, where you study a process at a certain scale, say the tissue scale, and then you take into account an organ scale and maybe a cellular scale. And then the time scale of the questions that you ask is exactly in the middle. It's the middle out. And that leads to complications, because then, can you assume the slow process to be constant? Well, probably not. So you really have to think about this transient behavior of the higher level scales. And that is really a hard modeling question. And when it comes down to computing, to simulations, that also makes it hard, because you have to iterate probably many times over the faster processes. And unfortunately, these fast processes are usually molecular, cell-based, whatever, are very expensive as compared to the processes that live on shorter timescales. And so you quickly get this combinatorial explosion of compute need. And that is one of the reasons why I didn't talk about it today, but that we were forced to start to push our models on these high performance computing machines. And maybe, I didn't say it, but maybe I should here also. So sometimes you find that your simulations don't fit in your laptop anymore, but let that never be a reason to stop. We even have a project called the CombioMed project, Computational Biomedicine project, which is supposed to help you. So if you run into computational problems because your models are so expensive or need so much memory that you basically give up, I would do one final thing and give a call to the people in this project, and they may be able to help you to push to the next level in cloud HPC or HPC or what have you. But we had to do that in these red blood cell models, in these agent-based models, in order to be able to do our simulations.
Yeah, yeah, definitely. That's a big issue, and I think that's also a good take-home message, saying that everyone who is working in this in this field of agent-based modeling has to be prepared at some point to get into the code, maybe to rewrite the code in C++ or in whatever, so that to make it compliant.
Today I really talked about the modeling aspects and the biomedical aspects of these models, but you're absolutely right. These are not models that you would easily implement in an ANSYS code or so. And so the flow part could be done by a commercial solver probably. We used a flow solver that we took basically from the internet, but it's based on the lattice-Boltzmann method, but that's a little detail. But the agent model was completely built in-house and completely developed from scratch. And that did require also, a few generation of computer scientists to set up the whole thing and to parallelize, to really bring it to the level that we can now run these simulations. And that is true. I mean, and that could be a showstopper. So if you really have to do those kinds of investments, but then, for instance, a group like mine in a computation science lab embedded in a computer science department helps. And maybe other people are embedded in more in biomedical types of departments where the computer science may be less developed. Well, hey, let's help each other. Those are the most interesting and fruitful projects that we team up and try to push the boundaries a little bit more.
Yeah, sure. So I don't know if we have questions. So we had a pretty intense day.
Yeah, I can imagine. No worries there. And it's late, eh? I'm in between you and a good lunch, or a good dinner, I guess.
Okay, so then maybe I think that if we, well, actually, I have a last question, if you permit me, Alphonse.
Sure.
Yeah, you know that in this summer school, we're talking about the kind of models you presented, this physiology-based models, etc. But actually, the second day is dedicated to data, so there is actually a lot of data science, etc.
Yeah, good.
So now, what is the place actually of data science? So within the kind of modeling that you
Yeah, that is also a very good question. I mean, and so what I've presented today is purely based on what you could call white box modeling. So we take knowledge from biology, from physiology, and translate that in a mathematical model. And you could also take the other approach where you would take your data and then create data-driven models. And we now call that AI or machine learning based methods. I think we are quickly moving into a field where we're combining these approaches. For instance, what I didn't tell you is that, of course, once you have a validated computational model, that becomes a source of data. And you can use that in one way or the other, or you may combine that data with other data sources in order to better train machine learning models. And we've seen some very interesting examples there. What we also see a lot is that we use very advanced data-driven models to create surrogates, surrogate modeling, where the original mathematical model may be too expensive in terms of compute. So even on the biggest machines, it's still too expensive. I will give you an example in a minute. And then you would create a surrogate model. So that could be some convolutional neural network that you train on your simulation data. And then, of course, when you then use that convolutional neural network, you hardly need to compute. The training is expensive. When I showed you the example or the results of the uncertainty quantification for the in-stent restenosis, that was partly based on surrogate modeling, because it is just too expensive to run or to sample over 5, 6, 7 unknown parameters and then run explicit simulations of such a model. So we currently have a PhD student and a postdoc, Dongwei and Pavel Sun, working on creating surrogates of the in-stent restenosis for the purpose of the uncertainty quantification. Otherwise, it becomes computationally intractable. But I believe that data science is here to stay. It is fantastic technology. We have so much data out there that we can use. So the hard part of that is actually, getting the data in a form that you can actually use it for the work that you do. And as I said before, the reusing of data and I think we should really dedicate more sufficient or lots of effort to that. But then we enter very interesting options of using both simulated data and observational data in hybrid gray box modeling environments. And I'm absolutely sure that we're just scratching the surface there right now, that there are many, many interesting opportunities.
Certainly. Okay. So I think that maybe actually it's 20 to 7. So 20 past 7, sorry. I'm tired too. Okay. Alfonso, thank you again for
Thank you very much for having me. I wish you a wonderful evening and dinner and a very successful next, I think 3 days, right? Or two more days in the workshop.
Yeah, 2 more days. Yeah.
2 more days. So good luck.